Hey, welcome to the little snapshot of DNA structure replication. We'll try and wrap this up in about 5 minutes. This is for your review, but we want to start with review and background. And just remember that proteins determine your traits because they are the ones that determine the structure of the cells and form enzymes and control the reactions and hormones and all that. So proteins are really important, but which proteins are made really depends on what the code of the DNA is. So the genes that are located on the chromosomes are made from DNA, and whatever code that is, that's going to make our proteins. So if we look at the DNA structure, the DNA molecule is made up of what are called nucleotides. A bunch of nucleotides makes a DNA molecule, and what nucleotides are made up of? A sugar, a phosphate, and a base. And the four bases are adenine and thymine and cytosine and guanine. And just like a puzzle, they match up with each other. So A pairs with T, and C pairs with G. Cytosine and guanine match up, and because they match up with each other, they are called complementary bases. So if we look at the nitrogenous bases, they're connected to the sides by the sugar and phosphate, and they form a ladder-like structure. So we have our deoxyribose sugar, we have our phosphate, we have our base in the middle, and that is one nucleotide. You put a whole bunch of nucleotides together, and you get your DNA molecule. So this structure is twisted to form what's called a double helix. It's like a spiral staircase. So when we use terms like DNA copying itself or chromosome copying itself, we really mean DNA replication. And again, remember that this happens in mitosis and meiosis. Chromosomes copying means that DNA is replicating. So also remember that chromosomes are made of DNA. So again, copying chromosomes means copying DNA. So 2 to 3 overall steps, pretty easy. Right, the first is that DNA has to unzip itself, and that's used or it's unzipped by a molecule called DNA helicase. So it unzips, and then the complementary base pairs, they're called free floaters, start to match up with one another. So here we go. We have helicase that's just going to unzip this DNA molecule, and these free floaters over here are going to match up with their base pair. They're just floating in the cytoplasm, waiting to match up with their pair. All right. So we have our free floaters, and then we have on the outside the DNA molecule is unzipped. So as the helicase continues to unzip, another enzyme called DNA polymerase is actually going to make sure that these are connected correctly. So we have the helicase unzips, the DNA polymerase makes sure that everything is matched up correctly. If not, they're going to kick them out and bring them back in. Make sure that A's and T's are paired together and C's and G's are paired together. If not, we get what's called a mutation. And that could be helpful, that could be harmful, or that could have no effect. A lot of times mutations don't have a whole lot of effect, but some that are harmful are really harmful. They can be fatal. So the result again is two new strands that are identical to one another. So when we say DNA is copied or DNA or chromosomes are copied, we really mean DNA is replicating each other. So there you have it. That is the end of the DNA replication review.